Okay, good morning, everyone. It's a pleasure to be here. My name is Natalia Matveyeva-Alexeyevich. I'm the head of Plant Sciences Core Facilities. So I will describe briefly what our core facility is about. Yeah, okay, that's good. So our core facility offers to academic institutions and industrial partners access to the research infrastructure for basic growth, environmental simulation, and phenotyping analysis. Also, our facility operates 15 phytotrons, 10 greenhouses, for small growth chambers like personals or fungi, and also one plant screen phenotyping system. Our core facility serves for 7 research groups at SciTech MU. It's about 70 researchers, postdocs, and PhD students. And the same as all other core facilities at SciTech MU, we are working based on the user fees. As I know, there are no excursions planned for the core facilities today. So for those of you who have never been in our facilities, I add just a couple of photos of our growth chambers so that you would so that you could imagine how our growth chambers look inside. So this is one of the type of phytotrons that we have. It's open shell phytotron, where the illumination has each shell, the temperature is the same, and the humidity is the same for the whole chamber. So this type of phytotrons we are using for the basic plant growth. Also, this is another type of growth chamber, the phytotron with the closed vent, where each shell is a mini phytotron where it mixes out totally different conditions. For example, the temperature could be different among next shells for up to 10 degrees. So within one phytotron, we could run up to 12 different experiments at the same time, which is quite convenient for the research purposes. We also have greenhouses with either LED light panels or high pressure sodium lamps for the experiment. We are able to control the following parameters. This is temperature, humidity, light intensity, day and night length. For our plants, we could simulate the sunrise and sunset to make it more close to the nature. And also in some chambers, we could adjust the various gas conditions, such as ethylene or CO2. There are several types of indoor illumination available at our growth chambers, such as fluorescent lamps, high-pressure sodium lamps, LED panels. We have different types of LED panels, starting from the standard one, where the combination of cold white, red, and far-red lights are. We also have highlight LED panels and multi-channel LED panels, where up to nine wavelengths can be combined and adjusted. The simulation of the following environmental stress conditions can possible to provide in our facility. Or we could simulate the cold stress, the heat stress, the highlight stress, and drought stress for the experimental plants. We also have a plant screen system delivered by Photon System Instrument. This is a high precision, low throughput digital plant phenotyping for small and mid-sized scale plants, such as Arabidopsis, tobacco, maybe early stages of brassica. So the total height of the experimental plants must not be higher than 1.2 meters. Everything that is below is possible to use for the experiment and for measurements with our plant screen system. This system equipped with sensors for RGB and kinetic chlorophyll fluorescence imaging, and also contains an integrated root model, which allowed us to investigate a root architecture of experimental plants. So basically, we have one system for 3 different types of experiments: for morphometric imaging, for chlorophyll fluorescence imaging, and for root phenotyping. I will briefly describe each type of experiment that is possible to measure with this system. So when we are talking about the morphometric imaging, we can measure a list of parameters that otherwise would be very difficult to measure manually in a very short and effective way with the RGB digital imaging, such as area, perimeter, compactness, roundness, etc. When we are talking about the plant phenotyping of the kinetic chlorophyll fluorescence imaging, so this imaging is used for rapid, non-invasive measurement of photosystem 2 activity. As you know, fluorescence technique is used as a rapid indicator of photosynthetic performance of plants in different developmental stages and in response to changing environment. So the advantage of this imaging over the other methods for monitoring plant development and stress is that changes in chlorophyll kinetic parameters often occur well before visible changes are apparent. So, and the last type of experiment that you could measure with our phenotyping system is the root model imaging. So we can make high-resolution images which allow us to visualize and do a morphometric analysis of the visible root system with automated measurement of various parameters related to root dimension and morphology. So we have a number of rhizotrons that are imaged by the high-resolution monochromatic camera, and under the homogeneous LED light source illumination, the root is imaged via the transparent side of the rhizotron box, which give us very good pictures of the root architecture of our plant. Our rhizotrons are quite compact, let's say so they will not, it is not possible to use them for plants with a huge root system, but for those which has a root up to, let's say, 40 centimeter length, it's possible to use. So it serves well for Arabidopsis and other smaller plants. But for bigger plants, like for example, Brassica, it is possible to use it only at the very early stages of the development of root system. So this is about our plant phenotyping system, which we could offer for our users. We also have a biostatistician who could help our users with the statistical analysis of the phenotyping data. So we have our colleague Ingrid Kovacheva, who is a biostatistician and who could help our users with analysis of different type of plant data, such as phenotypic data, chemical kinetic fluorescence, chlorophyll data, and so on and so on. For those of you who are already our users, I must remind you to do not forget to acknowledge the core facility when you prepare the publications with the data that were obtained with the help of the core facilities. This is extremely important for us, so please don't forget to acknowledge the core facility if you use it for receiving the experimental data. Basically, this is all what I wanted to share with you today. If you have any questions or requests, feel free to contact us via the email. We will be happy to collaborate. Thank you. Please do have questions because there is plenty of time left. We have still 8 minutes, so if you have questions for Natalia, don't hesitate to ask.
Okay, I have the gas concentration. So you were mentioned that you could control the carbon dioxide. And what range of concentration can you use? And is it possible to mix with other gases like nitrogen or?
Unfortunately not, we have only one source CO2. Either. The mix of gases, I'm not sure which you can use because The system is designed the way that we will get this gas inside the first well, and then some absorbent should absorb it. If we will add two different gases, I'm not sure whether the second gas will not go to the room. So it's not very key, let's say. So, but I could imagine that instead of CO2, we probably could use nitrogen, but we
Okay,
never
so-
did it so far. So.
It's with the atmospheric air, and then you found only the one.
Yes. And the exact numbers I will probably not tell you, but what we did experimentally, it's 2 or 3 times more than the CO2 we have in the air normally. That was the experiments we did, and our system worked with that. Quite okay. In case if we need to run the experiments with a higher concentration of gas, we would test the system first and then start be sure that the instrument is capable to hold this level of gas.
Okay, thank you.
If I can just out of curiosity, do you work in with a relatively sterile environment, or do you somehow control the microbiota of the soil when growing the plants?
Good question. I wish we could work in a sterile condition, but it's not. Because we trust our supplier of our soil. Theoretically, they should sterilize the soil, but we never tested this. And those facilities who want to do it in a very, very sterile way, it's possible to autoclave the soil, but then we would kill all the positive microbiome, components. So basically, we just carefully choose the supplier of the substrate to be sure that it does not contain any fungi, let's say so. Or we have a protocols how to fight with fungi, but not to kill something that is helpful for our plants. So this is the way how we
Do you, maybe I've missed that, do you also do some imaging, like microscopic imaging directly inside the greenhouses? Is it possible?
like microscope, we have a microscopic facility.
Yeah.
We have a simple binocular microscope that helps us to do process, but it's not really the microscope. It's just to help us to manipulate with the flowers and to do the process, but otherwise.
Nadia, could you speak up? It's
Yeah.
difficult to hear in the back.
Yeah, I'm very sorry. So we have a very simple binocular microscopy which microscopes that help us to do process, but otherwise for the more complicated, for the microscopic analysis, we have a core facility that has a very advanced microscopes, and we are at the same floor, so it's basically next to our core facility, which is very convenient. For us, the plant could be taking and do the microscopic analysis at the same corridor, let's say so. So otherwise, no, we don't have an advanced microscope at the core facility.
And the phenotypic station is based on microscope, no? Or is it?
No, because the plant is big enough to make pictures like it is. It's RGB camera that make photos, but the plant is big enough to make just without any,
But do you have some lens there, or is there some?
We could put it lower and higher. This way I can make this.
Okay.
Photos. We need a more, bigger picture.
So you cannot change the magnification and of the system.
A little bit, but not the same way as you could do it with your microscope.
There's still time for you to highlight something, just for 2 minutes. Like, is there anything else you'd like to share with the audience?
Okay. Okay.
Yes, please. I was also curious about the day-night cycle you mentioned.
Yes.
And do you only control for light conditions or other conditions as well? Like temperature changes, humidity, stuff like that? Or is it just light?
Temperature for sure. We could change it in a very big range. From a cold stress, it's +4 to the heat stress, it's +40 degrees. This is the range of changes that we could use for the different experiments. The humidity, I would not say that we are changing it significantly. Our aim is to keep it on the same level, about 50% humidity, which is quite comfortable humidity level for the plants. But the light intensity, the light quality, the light spectrum, this is what we are usually use for different types of experiments. It's a long day, short day. With the multi-channel banks, it's possible to basically design your own light spectrum that you want to provide to your experimental plants in case if the standard LED panels are not effective enough. Or for example, our highlight LED panels were very useful when our collaborators brought plants from mountains and they wanted to grow them, but the light intensity in the standard growth banks were not enough for those plants, which used to grow very high in the mountains. So we used those highlight LED panels to provide them 10 times more light intensity than we usually do for our standard experiments. So yes, light and temperature, basically those two parameters that we could change significantly for our by request of our users.